Good morning, students. After completing protein and carbohydrate digestion, we will start the fat digestion. But before going into the process of fat digestion, we will first learn about bile because bile is the most important substance in fat digestion. So we have already learned what is bile, but then also we will just have a recap of what bile is actually. And after that, we will move on to the fat digestion process. Basically, it's a complex fluid which contains various substances. We know it's yellow in color and yellow, not proper yellow, yellowish green in color, and like this. The color is similar to this, slightly light like this, this color. Similar to this, and bitter in taste. Okay, and it has a different kind of substances with it which actually help in fat digestion. So today we'll be learning about the main function of bile in fat digestion. What does bile do in fat digestion? So first let us see the composition of bile. It has 97.6% water and 2.4% solid. Under this solid, you have the organic substances consisting of the bile salts, bile pigments, cholesterol, fatty acids, the phospholipid lecithin, and the mucin. And inorganic substances, this question comes like the sodium, calcium, potassium, chloride, and biocarbonates. So a question comes like what are the different organic substances present in bile? You have to mention the bile salts, bile pigments, and if the names of bile salts, bile pigments come, you have to mention that as well. And also what kind of inorganic substances are present in bile, you have to mention the sodium, calcium, potassium, chloride, and the biocarbon. So this is the basic composition of what bile is or what bile has in it. So you know that bile is secreted from the liver and stored in the gallbladder. You know that, right? So next we will be moving forward to the production of bile, which is very important. Why it is important? Because uh, from where does what are the ducts that is involved in the secretion of bile or the production of bile is very very important. So basically what happens key, it is a digestive enzyme. We all know it is produced by the liver, we all know that, and it is stored in the gallbladder, we know that. So this is the gallbladder, this is the gallbladder. So it is stored over here, and it is the duct is over here, the right hepatic means liver, so bile duct is here. So from here, this is a liver portion, and the whole thing is liver, and this is a duct, the hepatic bile duct. So from here it moves inside. Okay, it carried from the gallbladder to by the cystic duct. So here is the cystic duct. This portion is the cystic duct through which the bile comes. Okay, and enters the duodenum. You all know that bile enters into the duodenum. So here it is entering into the duodenum with the sphincter of Oddi. So this portion this portion is the sphincter of Oddi. So this is very very important, that from the gallbladder it is coming through the cystic duct. This is the cystic duct, and then it is moving to the duodenum or entering the duodenum with the help of this sphincter of Oddi. This is very very important. Okay, so you must know this, you must keep this in mind. That is, it is coming from the gallbladder through the cystic duct, and it is entering the duodenum through the sphincter of Oddi. Okay, now, there is a function of bile which is very, very important. That is the cholagogue action. Okay, we know it is a cholagogue action. So, what is this cholagogue action? Cholagogue action of bile is basically that it acts as own stimulant. That means bile, whenever it is required, it acts as its own stimulant. As I have told you, I think you remember, that whenever there is fat, our body can understand. So CCK, or this cholecystokinin, is stimulated. When the cholecystokinin is stimulated, bile understands that okay, there is fat in our diet, and I must be secreted. So bile is secreted on the other side. Okay, so it acts as own stimulant. It's known as the cholagogue function of the bile. Next comes the function of bile in fat digestion. So this point is very important. We all know that bile helps in emulsification. Now, what is this emulsification? What do you mean by this emulsification? Suppose this is the fat globule. So fat globule means fat globules always come in a lump. Okay, you don't have separated fat globule, and fat always have a tendency to what I'll say, the tendency to clog with one another. So it is a complete mass, we can say. Okay, so this is a fat globule. So all fat globule part, you have to just segregate those fat globules. So if you have a lump like this, you have to break into smaller particles so that the amylases can work on it. And this smaller particles, breaking into smaller particles, is mainly done by bile. So it helps in emulsification. Okay, so the bigger fat globules is broken into smaller fat globules. Now if this is a bile salt, it is done by a bile salt. Bile salt has a hydrophobic part, that means the part which fears of water, and a hydrophilic part, that parts which loves water. Okay, philic means love, phobic means fear. So this green balls over here, the green balls is the philic part, hydrophilic part, loving part, and the yellow ball over here, it is the phobic part, that is means the fearful part. So if this is a fat globule, you can see this blue ball over here. So you can just take this blue ball over here. So the phobic part is attached. As you know that the oil and the water can never mix with each other, right? So what happens, the phobic part is getting attached to the fat molecule. So this is the phobic part, it is getting attached to the fat molecules, and the philic part is opposite to the fat molecules, because water and fat doesn't mix. Okay, so the philic part is opposite to the fat molecule, and the phobic part is attached to the fat molecules, and they are moving as a small droplet. Okay, so these are the small droplets. So these are the emulsified one, the smaller one. So you can see this is a big fat droplet, fat globule, a bigger one, and these are the smaller fat globules on which basically the amylase enzyme, the pancreatic the pancreatic enzymes will be, not amylase, the pancreatic enzymes will be lipase enzyme, sorry, the pancreatic lipase enzyme will act on it. Okay, so let us check the first and the foremost and the main function of bile is this emulsification. Now, what are the functions related to this emulsification or after emulsification that the bile do, we will learn next. The next one is cholic acid activates lipase. So we know that they have bile salts, some of sodium taurocolate, colate word, it comes from this cholic acid. These are the cholic acid derivatives. Okay, cholic acid derivatives. Let me take another color. Okay, cholic acid derivatives. So all these bile salts, this bile salt will contain sodium taurocolate, sodium glycocolate. So this colate means cholic acid derivative. This cholic acid will activate the lipase present, and lipase means this lipase after activation will act upon this emulsified fat droplets. Okay, and they will digest the fat. Next is good solvent. Definitely emulsive, a big fat molecules which is a clump, it will not be mixed in a solvent, whereas this broken part, this emulsified fat, they will be a good solvent, they will mix very properly. Stimulates peristalsis. Whenever we are trying to digest, when this emulsified fat is formed and the lipase is acting, there is a peristaltic movement is very fast, and that is done by the bile. So passing out of the fat molecules with the help of bile, stimulating the peristaltic movement is done after this emulsification with the bile. as I already discussed, acts as own stimulant, the cholagogue function of the bile. Last but not the least, it maintains the pH. So it has an alkaline pH. So as I have discussed also previously, in the digestion of protein, if you remember that the HCl, the protein after coming out from the stomach, the bile maintains the pH and tend that thing to alkaline, so that the pancreatic amylase enzymes can actually act on it. Okay, the amylase can actually act. So the alkaline medium is maintained or changed from the acidic medium with the help of bile.